Have you ever kept checking your phone for reply, even after you already knew the conversation was over? Your brain had learned that checking the phone meant a reward, and it takes real effort to unlearn that. Today's paper asks a precise version of that question: which neurons in the brain actually pull the brakes on a learned behavior, and how do they do it?
Oh, that's such a relatable setup. So the paper actually identified the specific cells responsible.
Exactly, and trace the circuit all the way from the front of the brain down to a reward hub deep in the midbrain. It's a remarkably complete picture.
Let's start from the basics. What do neuroscientists mean when they talk about contingency degradation?
A contingency is the link between a cue and a reward. Think of it as a rule. When I hear this sound, food is coming. Contingency degradation is when that rule stops being true. The cue still appears, but the reward no longer reliably follows. The brain has to learn that the old rule is broken.
That sounds like it should be straightforward. Just stop expecting the reward.
You think so, but it turns out learning from repeated failures is surprisingly hard for the brain. Animals, including us, tend to stick with old strategies for quite a while after they stop working. We keep reaching for the metaphorical bowl even after it's been empty many times in a row.
So which part of the brain was already known to handle this?
The medial prefrontal cortex, called the MPFC, a region at the front of the brain involved in high-level decision-making. Research going back decades shows that when the MPFC is damaged in rodents, they become what neuroscientists call perseverative. They keep responding to a cue long after it has stopped predicting reward. They simply cannot let go of the old habit.
So the MPFC acts like a brake on outdated habits.
That's a nice way to put it, but the specific neurons doing that braking and how they communicate with the broader reward system to actually change behavior, that remained a mystery. This paper set out to solve it.
And there's a connection to addiction here as well, right?
The authors raise it explicitly. In addiction, certain cues, a smell, a place, a social setting, become powerfully linked to the reward of a drug. Even after someone stops using, those cue associations can trigger relapse. Understanding how the brain normally degrades these links could point toward what goes wrong in addiction. The authors suggest the specific circuit they found may be weakened in people with active substance use disorders. Before we get to the experiments, I want to walk you through the theoretical model the team built, because it underpins everything else. You may have heard of reinforcement learning, the idea that the brain learns by tracking what are called reward prediction errors.
Is a reward prediction error the gap between what you expected and what you've actually got?
Precisely. If you expected a reward and received one, the error is small. If you expected a reward and got nothing, you get a large negative error. Classic learning models say the brain uses these errors trial by trial to update how much it values a cue.
So what was wrong with that standard model here?
When mice went through a reversal, when the cue stopped predicting reward, their behavior did not change right away, even though prediction errors were already large from the very first omission. Animals kept licking in anticipation of the now worthless cue for many more trials before they started to change. The standard model would predict biggest error, fastest change. The mice did the opposite.
It's like they needed a string of failures before they would accept the situation.
Exactly the insight. The researchers proposed that the brain doesn't just use single trial errors, it also computes a running average of those errors over multiple trials. They called this the meta-RPE, or MRPE for short. Think of it like a slow-building alarm that only sounds after enough repeated failures have piled up.
So the MRPE peaks in the middle of the reversal, when failures have been accumulating, but the animal hasn't fully updated yet.
Precisely. The model separates two directions: a contingency degradation signal, abbreviated CD, which tracks accumulated negative errors, and a contingency enhancement signal, C, for accumulated positive ones. The CD signal is what tells the brain this cue is losing value, and CE does the reverse for a cue that is suddenly becoming rewarding.
And this new model fit the mice's actual behavior better than older alternatives.
Significantly better, even after applying a penalty for using more parameters. The team tested it against 5 competing models, including well-known learning models with dynamic rates. The Meta-RPE model outperformed all of them, and it was the only one that correctly predicted the timing of when animals would begin to change their behavior. Now, how did they test whether this model actually corresponds to real brain activity? The main technique was two-photon calcium imaging, a method that lets you watch individual neurons in a living, behaving mouse in real time across many weeks.
How does that work in practice?
Neurons are genetically engineered to produce a fluorescent protein called GCAR-MP6, is a sensor that glows brighter when calcium floods into a cell, which happens when the neuron fires. A two-photon microscope uses near-infrared laser light to detect that glow deep in living tissue. The team implanted tiny glass prisms called microprisms into the MPFC to create a clear optical window. And they tracked nearly 12,000 neurons over multiple weeks of the reversal learning experiment.
That scale of tracking across weeks is remarkable. How did they figure out which neurons were specifically responding to the contingency degradation signal rather than just to the cue itself?
That's where a statistical tool called a generalized linear model, or GLM, came in. For each neuron, the GLM asks, does this cell's activity over time match the modeled CD signal more than you'd expect by random chance? Because neural activity and task events unfold together in time. They used a technique called circular shifting, repeatedly scrambling the timing of the neural data relative to the task to build a realistic chance baseline, then compared the real alignment against that baseline.
And there was an optogenetics component too.
Yes, and this is where the study becomes especially powerful. Imaging alone only tells you about a correlation. To prove that CD cells actually cause behavior to change, the team used single-cell holographic optogenetics. They introduced a light-sensitive protein called CHRMine into MPFC neurons, a device called a spatial light modulator. Think of it as a precision projector targeting individual cells with sculpted beams of light, was used to activate the specific CD encoding neurons identified during the imaging phase.
So they could switch on exactly those neurons on specific trials, while the mouse was doing the task.
Exactly, they ran 3 separate versions of this experiment. In each version, the stimulated CD cells were compared against a different control group, unstimulated trials, neurons that encoded nothing relevant to the task, and neurons that encoded the cue identity, but not CD. That layered design let them attribute any behavioral effect specifically to the CD cell activation. They also used fiber photometry to track the connection between MPFC and a region called the ventral tegmental area, or VTA, a hub in the midbrain that drives motivation and reward through the release of dopamine. A thin optical fiber placed near the VTA or in the MPFC recorded population-level dopamine signals across the reversal.
And they also tested what happens when you block dopamine from reaching the MPFC.
Right, they infused a cocktail of drugs that block dopamine receptors directly into the MPFC while recording the MPFC to VTA pathway signals. That pharmacological manipulation let them test causality from the dopamine side of the loop. So what did they find? Starting with the imaging, about 13% of MPFC neurons showed significant encoding of the CD signal. That's a dedicated subpopulation, roughly 1 in 8 cells, active in a pattern that mirrors the modeled contingency degradation signal across the reversal, and the signal was stable over days.
Were those cells separate from neurons that responded to the cue or to reward value?
Largely, yes. The overlap with cue encoding or value encoding cells was far smaller than expected by chance. Most CD cells carried the CD signal and essentially nothing else from the task. Within those cells, about 64% were excited during the reversal and 36% were inhibited, but their average activity faithfully tracked the CD model signal.
And what happened when they activated those cells with the light-based approach?
The mice licked significantly less in anticipation of the formerly high-value cue across all three control experiments. When the researchers fit the MRPE model to the stimulation day behavior, the estimated CD amplitude was significantly larger on the stimulated trials, meaning the brain was acting as if the cue had lost even more value than it actually had.
So artificially activating the CD cells made the mice treat a cue as less rewarding. That's a clean causal result.
And the effect was lasting. When they checked sessions after the active stimulation period ended, the licking had not bounced back to pre-reversal levels. The mice had genuinely learned that the cue was less valuable. The activation hadn't just temporarily suppressed their licking.
What did the recordings of the MPFC to VTA connection show?
Multiple converging findings. First, dopamine released in the MPFC, arriving from the VTA, was found to encode positive reward prediction errors, particularly during contingency enhancement when a low-value cue suddenly starts predicting reward. This is consistent with VTA dopamine carrying the raw single-trial error signal up to the MPFC, so the MPFC can compute its running average.
And then the MPFC sends the CD signal back down to the VTA.
That's what the imaging showed. When they specifically recorded only MPFC neurons that project to the VTA, identified by injecting a labeling virus into the VTA, about 62% of those projection neurons encoded CD. Compare that to 13% in the general MPFC population. The pathway going from MPFC to VTA is strongly enriched for this devaluation signal.
And blocking dopamine in the MPFC slowed down the reversal.
Significantly, animals treated with the dopamine receptor blockers during the reversal showed a delayed midpoint of the behavioral change and a reduced CD amplitude in the model. The CD encoding in the MPFC to VTA terminal signals also dropped. Conversely, optogenetically activating the MPFC to VTA pathway during the reversal sped it up. Both the start and the midpoint of behavioral updating arrived significantly earlier in those animals.
Let me try to assemble the full circuit. The VTA sends dopamine up to the MPFC, encoding single-trial prediction errors. The MPFC averages those errors across trials into the CD signal. The MPFC then sends CD back down to the VTA. But what does the VTA actually do with that return input?
That's the final piece, and the team addressed it with ex vivo experiments, imaging in brain slices rather than a living animal. The VTA contains two important neuron types. There are dopaminergic neurons, the ones that release dopamine and signal positive predictions. And there are GABAergic neurons. GABA is the brain's main inhibitory messenger, which can silence the dopamine neurons, effectively generating a negative prediction error when they fire.
So if the MPFC to VTA input excites the GABA neurons, that suppresses dopamine release, which looks like the reward was less than expected. A bigger negative error.
That's exactly what the data showed. Stimulating the MPFC inputs to VTA in brain slices activated about 50% of the GABA neurons, mostly through excitation. The dopamine neurons showed a smaller and more mixed response. About 36% responded, split roughly evenly between excitation and inhibition.
So the MPFC is amplifying negative prediction errors in the VTA, which drives the brain to further lower its valuation of the cue.
That's the core mechanism. The MetaRPE model says CD increases the gain of omission-related prediction errors. The biology says MPFC sends its CD signal to VTA GABA neurons, which suppress dopamine, which signals a larger negative error than the raw omission would have produced alone. It's a feedback loop that accelerates devaluation learning.
And the mix of excited and inhibited CD cells in the MPFC reflects the heterogeneous responses in the VTA targets. It all fits together.
That internal consistency is one of the most satisfying things about this paper. Each layer of the model has a corresponding circuit element. As for broader implications, the authors note that distinct circuit elements appear to handle learning from gains versus learning from losses. Dopamine arriving in the MPFC seem to primarily carry the positive CE signal, while the MPFC to VTA projection neurons carried the negative CD signal. Cognitive flexibility depends on both arms working in balance. The authors also note that hypofrontality, the reduced prefrontal activity seen in active substance use disorder, could specifically impair this CD encoding, leaving drug-associated cues with inflated value that resists normal degradation. That's speculative, but it's a testable and clinically meaningful prediction.
If I had to put today's biggest surprise in one sentence, the brain keeps a running tally of your failures with a cue, and only after that tally crosses a threshold does a dedicated circuit in the prefrontal cortex send a signal to the dopamine system to start writing down a lower value for that cue.
That's exactly it, and neither region can do the job alone. The VTA provides the raw error signal trial by trial. The MPFC integrates it into a devaluation signal and sends it back. Disrupting either half of that loop, blocking dopamine, or silencing the projection neurons stalls the reversal.
The fact that you can speed up forgetting an old habit by activating that pathway with light is a remarkable demonstration of what targeted circuit control might look like one day.
The authors would likely agree. Today's paper is by H. Jort and colleagues, published in Nature in 2026, titled Prefrontal to Ventral Tegmental Area Dynamics Drive Contingency Degradation. DOI and all references are in the show notes. Thanks for joining us on OSCI Pod.
See you next time.